Okay, so today we will study the lecture 2, the rest of the part of the world of viruses. And okay, so in COVID-19, 6.5 million deaths worldwide, and as of September 26, 2022. So that was a pandemic, and people typically are afraid when they don't understand something, and in this pandemic, people want to blame others and spread frightening news in Virology reporting, like don't go to the UN and don't leave Wuhan, and coronavirus could mutate and spread further. China officials warns, and China warns virus could mutate and spread as death toll rises. Evidences has shown that the diseases has been transmitted through the respiratory tract, and there is a possibility of viral mutations. National Health Commission's Vice Minister said this one. And another news article emphasis on the coronavirus is mutating and Americans leaders are flying blind. So they presented the news as the mutation of the coronavirus is a surprising thing. Actually, this is not. It's very common and it's very normal for all the virus. So the virus is always mutating, and at every reproduction cycle and every cell in our bodies that's infected. And the question is whether it means anything. Actually, the answer is in the study of the origin of viruses. Okay, so the classical hypothesis, there are some classical hypotheses and which are based on the fact that the viruses are ancient and viral protein were existed before the divergence of the life, and thus infecting the last universal common ancestor. And this indicates that some viruses emerge early in the evolution of the life. So লাইফের ইভলিউশন হওয়ার আগেই ভাইরাসগুলো ইনফেক্ট করেছিল. সো নিউ গ্রুপস অফ ভাইরাস হ্যাভ রিপিটেডলি বিকাম ভিজিবল অ্যাট দ্য স্টেজ অফ ইভলিউশন, and often through the displacement of ancestral structure and genome replication chains. So one of the problems for studying viral origin and evolution is the high rate of viral mutations, particularly in the case of RNA viruses like COVID or HIV/AIDS. And there are three classical hypotheses on the origin of the virus. The first one is viral hypothesis. So here the virus evolved from complex molecules of protein and nucleic acid before cell first appeared on the earth, and indicated that virus contributed to the series of cell life. And this hypothesis has been supported by the idea that all viral genomes encode proteins that do not have cellular homologous, and this hypothesis is dismissed by the violation of the definition of viruses, in that virus require a host cell to replicate. And second is reduction hypothesis or degeneracy hypothesis. So viruses were once small cell that parasitized larger cell, and this hypothesis is supported by the discovery of gene viruses with similar genetic materials to parasitic bacteria. And this does not explain why even the smallest of cellular parasites do not resemble viruses in any way. And the third hypothesis was escape hypothesis or vagrancy hypothesis, and it is thought that some viruses evolve from bits of DNA or RNA that escape from the genes or larger organism, and it does not explain the structure that are unique to virus and are not seen anywhere in cell, and does not explain the complex capsids and other structure of viral particles. So the evolution of the virus. What is the evolution? The genomes of many organisms contains endogenous viral elements, and these DNA sequences are the remnants of ancient viral gene. Like the genomes of most vertebrate species contains 100 to 1000 of sequences derived from ancient retrovirus, and this is a valuable source to study the evolutionary history of viruses. And the viruses develop through changes in their DNA or RNA, and quite rapidly, and the best adapted mutants survive. The RNA viruses are more susceptible to mutants, and there is no mechanism to correct RNA in the host cell. In just one cycle of replication, one virus particles can produce millions of progeny viruses. Therefore, few defective viruses is not a problem. Most mutations are silent but can give advantages to viruses that increases their fitness, like changes that makes a virus unidentified by the immune system or antiviral drugs less effective. Both of these changes occur frequently with HIV. Many viruses, like influenza viruses, can shuffle their genes with other viruses. When two similar strains infect the same cell, results appearing new and more virulent strains. A group of closely related but distinct viruses can infect a broad range of animals. It is not possible to prove which of these rapidly evolving virus is earliest, but a closely related group of viruses in diverse hosts suggested that the possibility that their common ancestor is ancient. So, what are the ancient references of viral disease? So, this mummy shows that the postural lesions on the face are similar to those result from smallpox lesion. And in the 15th century, the Chinese practiced variolation, a method of nasal insufflation administered by blowing powdered smallpox materials provided from nose strip scabs of patients with mild smallpox. And there was no knowledge of causing agent, but simply that the survivors were protected. And in 17th century, Lady Mary, wife of British ambassador to Turkey, noticed this process, and she brought it back to the UK, and it's spread. In 17th century, Edward Jenner in England established the vaccination, and from then on is history. So, what is the concept of microorganism? Concept of microorganism developed from discoveries of three individuals at different times. Louis Pasteur conducted experiments on the relationship between germ and diseases, and Robert Koch discovers the field of medical bacteriology. And uh, no viruses identified yet, since the technology was not good enough. And however, in the literatures as early as 1728, viruses were used to describe as an agent that causes disease. Latin words virus meaning poisons, or so thought to be liquid or pastures. Every virus is microbe. So there are three results, or there are three statements were prevalent on those time. And key events in the virus discovery: in 1933, Helmut Roska built the first electron microscope in Germany, and he took the first electron micrograph of bacterial phage in 1939. He found that the filterable viruses were not liquid, and they were particles, and now they are named viruses, particulate agent. Okay. And key events in viral discovery, like key 1939 experiment provide that viruses were not simply small bacteria. Bacteria in broad culture begin to divide immediately by simply binary fusion. And viruses in broad, nothing happened, but um, in host cell they had the escape period, and thus did not divide by the binary fusion, a early different way of producing. And now we know that during explicit period, viral parts are being made, and then they are assembled, and the infectivity and then they are assembled, and the infectivity happens. Nowadays we know the 3D structure of many viruses. So, what are the reasons of studying virus? Virus discovery has always been driven only by disease, and some viruses causes disease. In humans, it ranges from the trivial to lethal, and also plays a role in the development of several types of cancer. Viral diseases can also affect the well-being of societies, and veterinary virology and plant virology also important because of the economic impact. So, foot and mouth viruses in domestic animals, rice yellow mottle viruses, and fuzz of lactic acid bacteria. Those are the examples. Some viruses are useful. First, typing of bacteria. Identification of first types of bacterial isolates provide epidemiological information during outbreaks. And viral sources of enzyme: reverse transcriptase from retroviruses and RNA polymerases from phages. Pesticides: some insect pests are controlled with baculoviruses and myxomavirus. And before discovery of antibiotic, phages, the viruses that infect bacteria, were used to treat some bacterial infections. A renewed interest to treat the antibiotic resistant strain of bacteria. Anti-cancer agent. Genetically modified herpes simplex virus and vaccinia virus are being investigated to infect and destroy a specific tumor cell. Certain baculovirus and adenoviruses are used as a vector to take genes into animal cell growing in culture for mass protein production. And gene vector for treatment of genetic disease. Like children with severe combined immunodeficiency have been successfully treated using retroviruses as vectors to introduce a non-mutated copy of the mutated gene responsible for the disease into their stem cell. Viruses studies have contributed to the knowledges. Much of the basic knowledges of molecular biology, cell biology, and cancer has been derived from studies with viruses. Here are a few examples, like a famous experiment carried by Alfred Hershey in 1952 used first T2 and E. coli to provide strong evidence that genes are composed of DNA. And the first evidence of first enhancer and transcription factor to be characterized were in gene of simian virus protein. Introns were discovered during studies of adenovirus transcriptions. The role of cap structure at the 5' end of eukaryotic mRNAs was discovered during studies with vaccinia virus and reoviruses. So viruses are small particles, and a concentrated suspension of virions might contain 10 to the power 12 virions per ml. A single virus infected cell might produce 10 to the power 5 virions. A person infected with HIV might produce 10 to the power 11 virions in a day. A virion is a complete functional virus that can infect living tissues. Virion is not cell, it does not contain organelles, and a virus includes genetic materials, capsid, envelope, and membrane proteins that allows virions to be to bind to its host cell to inject its DNA, RNA for replication. A virus will not have an envelope within the cell. If the cell is burst artificially, then virus particles are not virion because they lack certain proteins that make them infectious even though the genetic material is present. So viruses have gene. There are four possibilities of a viral genome. One is double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, and generally viral genomes are much smaller than host cell genome. And viruses encode all requirement for producing multiple new virions in a number of ways. This include virus uses host cell proteins. However, an RNA virus must encode RNA polymerase because host cell do not encode enzymes that replicate virus RNA. Virus code efficiently. Virus may have overlapping genes and genes within the genes. Viral proteins are multifunctional. A virus protein may have several enzymatic activities. So viruses are parasites because a new virion is never formed directly from a pre-existing virion. New virions are formed by replication inside a host cell and involves the synthesis of components followed by their assembly into virions. So viruses dependent on their hosts for most of their requirements, including building blocking such as amino acid and nucleosides, protein synthesizing machineries, energies in the form of ATPs. A virus modifies the intracellular environment of its host to enhance its own replication processes. Production of new membranous structures, reduce expressions of host cell genes, enhancement of a host cell processes. In the photosynthetic bacterial host, some phages encode proteins that boost photosynthesis, result in enhancing the virus yield. So, what are the definition of virus? Viruses are distinct biological entities with the following properties. A virus is an infectious, obligate intracellular parasite. The genetic materials of a virus is either DNA or RNA. The genetic materials of a virus enters a host cell and directs the production of the building blocks. New virions are made in the host cell by assembly of these building blocks, and new virions produced in a host cell then transport the viral genetic materials into another host cell or organism to carry out another round of infections. So there is an ongoing debate as to whether viruses are living or not living. The views taken depends on how life is dependent. So there is an argument viruses are not living. So in order to consider it alive, an organism must have a genome and metabolism. It must also be able to reproduce and to compensate for changes in the external environment to maintain homeostasis. Viruses have genome, but they have no metabolism and do not generate their own energy. They are unable to reproduce independently and do not undergo cell division like living things. They completely disassemble after gaining entry into the cell and assemble new variants from its case. Are viruses living or non-living? So arguments: viruses are alive. Viruses are Viruses have genes, and when they infect cell, these genes are replicated. So in this sense, viruses are living. When viruses are outside their host cell, they exist as virus particles, which are inert and could be described as non-living, but viable bacterial spores are inert and are not considered to be living, non-living. So viruses are very different to cellular life forms. So are viruses living or non-living? So this is the definition, very famous definition, where it shows virus belongs to biology because they process a gene, replicate, evolve, and are adapted to particular host, biotic habitats, and ecological niches. However, they are non-living infectious entities that can be said at best to lead a kind of broad life. So you might form your own views as to whether viruses are living or non-living as you progress through this course. Okay, till then, goodbye.